Okay, and now for a series of two talks about one of the most ancient and largest machines in nature, and who is better to give an overview than Ada Yonath to study it best. Ada, please.
Thank you. So, good morning. I apologize for having a cold and having problems in talking, and also in seeing because I had a minor accident. But I hope I will perform well. And I want to tell you that the first lecture will be more about the ribosome itself, and the second will be more about ribosomal antibiotics and new ways to look at antibiotics problems. So ribosome is the machinery that translates the genetic code in the form of messenger RNA that is being transcribed from DNA, and there is nobody better to talk about it than Roger. And then messenger reaches the machinery it's called ribosome, and according to this, proteins are being made. Do I have to tell you that DNA is 4 letters language? And each triplet is coding for an amino acid, and there are 21 amino acids. Okay, so I skipped that. Ribosomes are universal in terms of their function. So they perform their main functions, the two main tasks that they have, decoding and peptide bond formation, in a similar way in all cells. If you ask my opinion, I think in the same way, but it was not shown yet fully, so similar. And this is regardless of the source of the cells, the evolution level of them, and the size. And even the ribosomes are evolving with time because they have to interact with more complicated cellular components. Anyway, there is a huge number of ribosomes that function in each cell. Mammalian cells may contain millions of ribosomes of molecular weight of about 4,000 Daltons, each ribosome. So in the liver, for instance, the richest cells for ribosomes, there could be 4 or 5 million ribosomes in a single cell, and they function all the time. Even in bacteria, they can contain up to 80 to 100,000 ribosomes in a single cell when bacteria grows. They act continuously. They can form 5 to 40 peptide bonds. Peptide bonds are the bonds connecting amino acids. I know you know that. In a single second, and they hardly make mistakes. So let's talk about mistakes. The mistakes that we as scientists can catch is between, it had to be 500 to 1 million, 1 to a million, very high fidelity. But that's not the only interesting point. The second interesting point is the speed. So I think I see here some people that were my teachers or my mates. When I was here in this university, I had to make a peptide bond in second year. This was an exercise. And I was a fast student, 6 hours for one bond. Now I understand that chemistry developed, and mainly in San Diego they are making new proteins so they can make one bond in 2 hours. Instead of talking much, we made a little video. How do I darken here? Anybody knows how to put off the lights? It was okay before the lecture. You know? Oh, wonderful. So we made a movie when the structures came. Structures came in the beginning of the millennium, 2000, 2001. So I collected whatever structures were available, 4 or 5 of them, and interpolate between them. Please give me dark. And made this very short video together with 2 students from Bezalel, the Art Academy. Since then, you will see here bacterial ribosomes. Since then, nobody complained, and it's in YouTube. I didn't put it, but somebody put it in YouTube, and nobody complained. So I think that still they can show what ribosomes does, of course, in a explanatory way. So the art students, told us that cells must be green. This is why the background is green. And they also say that they will decide who comes to whom. So they decided that messenger RNA comes to the ribosome. And I'm not sure that this is the way it is, but I hope you forgive me not knowing that. Also, ribosomes are made of two subunits. The small subunit is the position, it provides the site for peptide, for decoding, and the large subunit for peptide bond formation. So you will see it in the movie. Here's the small subunit, and the messenger comes, and have a look. Look, now. Choop, it took it. Now this was a very small motion on the movie, but the whole part, this part of the small subunit that is usually called the head, because the small subunit is called also the duck by the electron microscopists. This motion here means moving all this thing, and this thing has a molecular weight of 250,000 Daltons. So it's not a small motion, it's just small when you look at it from the outside. So now messenger is in its path between what is called head and the body in a neck, and it is being controlled by a non-ribosomal factor, this one, the blue one, which is initiation factor number 3. So in bacteria there are 3 initiation factors. In human it's much more complicated, or in mammalians. And initiation factor 3 monitors the positioning of the messenger RNA, of the first tRNA, and the association between the two subunits. So here it monitors the messenger RNA, messenger goes round, and now when everything is fine, the first tRNA can be brought by the second initiation factor. Sorry, I should have said that the protein bond, the peptide bond is being made between 2 amino acids that are associated, they are acylated to tRNA molecules. tRNA molecules are the carriers. There are 3 tRNA sites, A, P, and C. P is the peptidyl transferase site. That's where peptide bond is being made. A is for the newly brought amino acid, acetylated tRNA, and E is simply it's the exit. So here is tRNA in yellow, or orangish. What brings it in is initiation factor 2, and the amino acids, sorry, but they're all shown here in blue, although they should have been in many other colors. So the 2 initiation factors are monitoring this state, that everything is sitting in place, and then, only then, they can dissociate, and the large subunit can approach. It will meet with the small subunit in a minute by compatibility of their surfaces, and also make conformational changes that create bridges. Have a look here. Bridges are being created, and there are 13 like them in bacteria that keeps the whole ribosome really in place. Now there is a functioning ribosome, and we look at it from the solvent side. tRNAs are coming here with elongation factors. The piece that you see helping in and out is the large subunit. It has 2 arms for that, and below it is the small subunit where decoding happens at that time. So every time a peptide bond is being made here in the large subunit, which we took away, the messenger is advancing in a triplet. So now let's have a look just how the peptide bond is being made. Not only fast, but tRNA changes its orientation. And the newly born protein comes out through a tunnel, which you saw earlier, and now it is shown again in the large subunit, spans it, and when the protein is large enough, it comes out and folds alone or with chaperones. We won't talk about it now. The process continues until it finishes when there is a stop codon. And then instead of tRNA comes factors like recycling factor, release factor, and they allow the two subunits to dissociate the protein to come out and do whatever it needs. And the tRNAs look for a new job, so is messenger RNA. That's the whole story. Very simple. And took us and some others that joined us during the trip about 20 years. In my case, 20 years minus 2 weeks. We couldn't see in the movie is the ribosomes. אפשר קצת יותר חושך בבקשה? What you couldn't see in the movie is that ribosomes work as polysomes. The many ribosomes are reading the same messenger. And in this electron micrograph that surprisingly, it took me some time to find out who made it, but it's in the 70s, beginning of the 70s, 69, 70, they could see that protein comes out from the other side of the messenger, that there are two subunits, just amazing. But I brought it here not only by that, but also by showing that ribosomes function together. I also didn't show in the movie that we have the atomic structure. It's not only us, but several. What I show here is our small subunit. It's made like all ribosomes except for mitochondrials. It's made mainly of ribosomal RNA, which is the gray part here. In the small subunit, in bacteria, about 1,200 nucleotides, and it's called 16S. It's highly indicative of evolution, but we talk about it later. It sediments at 30S, and it has up to 21 different proteins. It means 20 or 21. The total molecular weight is 0.85 Mega Dalton, and this is where decoding happens. Large subunit with the same color code, sediments as 50S. It is responsible for peptide bond formation and something that I will talk about, mention at least once, nation chain elongation. It's not just making peptide bonds, it's a polymerase. And it protects it. Molecular weight is 1.5 Mega Dalton, 3,000 nucleotides, and up to 34 different proteins. Here there is a little bit more variability between 30 to 34. When one looks at the two-dimensional map of the bacterial ribosome, I show here the one that belongs to the bacteria that I talk about, that we determine the structure of, the Anococcus radiodurans, but it's very similar in all. It is of the large subunit. It is made of several domains. And in one domain, when one looks carefully, not at the two-dimensional representation that I show here, but at the structure, one finds out that there is an internal region here, the bluish, which is made more or less of two halves that are related to each other by pseudo-symmetry, pseudo-twofold symmetry. This line is the sort of the axis, and this is the rotation that should be. So if we want to look at it better in more detail, this is the pseudo-symmetrical region. It's sizable compared to other molecules, but it's only 118 nucleotides out of the 4,500. And what is specific about it is that the main chain, the fold, is symmetrical. There is no sequence symmetry, and I'll come back to it. This symmetrical region in the ribosome, this is the large subunit. I hope you could see the gray. This is the symmetrical region, the blue and the green, and I will keep blue and green for the part of it that accommodates the A site tRNA in blue and the green in the P site tRNA, like here. So here, this is the blue region and the green region. tRNA in the A site is in bluish, and the second one is in greenish. Peptide bond formation happens here in the middle of it. And the whole region is connected to the rest of the ribosome, to the two moving parts, the two hands that come in and out directly by extension of the RNA. So it can be, we think that it can serve in the modern ribosome as a signal transmission between all the active components, or mainly active components. So it tells the two hands in and out. This is in and this is out when peptide bond is being made. And it can tell the messenger to progress through the tRNAs and one of the bridges. So it's sort of announcing, now there is a peptide bond, you can go on to the next one, and so on. If we look at it from top, it has the same fold in all known structures. Here there are only 7 of them, eukaryotes and prokaryotes, and even mitochondria. But actually, now there are many more structures. In all of them, it's exactly the same. So what I did here, the A site, the blue region, I put one on top of the other. You have here the table showing which one is which, but I cannot. They are actually the same, exactly the same, and the brain are the same too. Backbone, only backbone. So it's Eubacteria, Archaea, Eukaryotes, and mitochondria. And in the ribosome, it sits here, within the large subunit. And the rotation, this is a symmetry rotation between the two of them is around this point, which is the center, but also peptide bond formation center. And from the other side, here we look at it from top, but from the side, if we put the green on the blue or the blue on the green, as you want to see it, you see that the identity is very high, but not perfect. There are some changes here and there, but not around the peptide bond formation position. So just about peptide bond formation, for those that forgot their chemistry, these are 2 amino acids that make a peptide bond, and one water molecule goes out. In ribosome, it's a bit different, because they both come either as acylated tRNA or peptidylated tRNA. The water has gone earlier. So the bond is between, of course, NH2 and CO, but there is no water molecule that comes out. And that's really important, because for years, I myself was looking, where will water go? There is no space for it, there is no reason. So water has gone before. And this is the position of peptide bond formation. I'll show it again. Between the A site tRNA and the P site tRNA, the acetylated and the peptidylated, here, this will be the bond. And the newly bound protein will go to the tunnel. If we look at the peptide bond formation position from top, the fold is shown here. You can see some symmetry here, some pseudo-symmetry. It's because we cannot really cut it exactly at the symmetry, but almost. And when we put all the side chains, all the nucleotides, and peptide bond position, here it is being made. This will be the A site tRNA, the P site tRNA. You can see that the symmetry, the pseudo-symmetry, is kept also in the orientation of the nucleotides, not in their sequence. So this, for instance, is symmetrical to this. The pink here is symmetrical to the pink, and so on. One exception is this one, 2602, that reaches the position of peptide bond. The rest are on the rims and more or less keep the orientation and the symmetry. Have a look here, for instance. This is really very striking, that the orientation is the same, no sequence. So if we look at the peptide bond formation position, it's called PTC. It is here at the lower part of this cavity. It's made mostly of RNA, actually fully of RNA, because the only protein that sits here reaches 18 angstroms away from peptide bond formation. It means chemically it doesn't make any difference. So we put in crystallographically, the end of the tRNA that should go to the A site, and it's called tRNA acceptor stem mimic. Acceptor stem mimic sits here. And it really sits very nicely. This is the part of tRNA, and this is a CCA amino acid, CCA amino acid. We have to remember that the tRNA molecule is made of L-shaped double helical structure with the anticodon loop at its end, and the CCA amino acid, which I just showed, at the other end. Except for these two regions, it's double helical. Only here and here it's not. And that's very important to understand how it serves its various functions. So the motion of tRNA is shift, as almost every textbook shows, for the whole part until it reaches the CCA amino acid. This does not move, this rotates. And in the movie you could see it slightly, I will show it a bit more. So what I mean here, this is the acceptor stem of the A and the P in blue and green. All moves sideways, but this part rotates. And how does it rotate? It rotates around within a scaffold, ribosome scaffold, which is shown here. So these ribs are actually ribosome. And this is the A and the P site tRNAs, and this is the rotation motion. So this what I show here, inside here, I can show in more detail, more structural detail. So the volume that is occupied by A to P side rotation is shown here from blue to green. And the ribosome is the gray, the RNA around it. So actually what we see, if we look at it from top, A is on this side, P is on that side. You remember the nucleotide that bulged into peptide bond formation, and there is one on top of it. This is the orange. The one that is on the spot is the pinky, or I think you see it maybe purple. And the motion is in this direction, from A to P. Only of the small day, single chain CCA. The rest is moving sideways. So what happens? This we call the PTC rear wall in all types of brown. From A to P, this is the motion. This is the rotation. And there are two nucleotides in the middle that sit there. And if we want to look at it really carefully, it goes from A to P. We highlight here the pieces, the RNA part of the peptide bond formation, PTC, that are interacting with the rotating motion, with the rotating moiety. So here at the end, it comes to make the peptide bond. So actually, what we study from this is that the ribosome positions the 3' ends of the A and P site tRNAs in proper stereochemistry. Nothing else. Just put them there. The reaction is exothermic, and it's done by itself. So you can imagine that this was not completely accepted by all ribosomologists. When they all thought that hydrogen must be there, or proton, or something like this. So with time, we got some validations, verifications of our suggestion. Sato in Tokyo showed that there are some nucleotides that are essential for peptide bond formation in the PTC, and some that can be replaced. A nucleotide is needed but can be replaced. These are the yellows. And exactly he did know about our work, exactly in accord with what I showed you rotating, those that are highlighted. Venki Ramakrishnan published two structures with tRNA at the A site and the P site, and look how nice all these structures together with our derived one agree to each other. And Massa and Carle calculated the energy molecular and energetical quantum potentials, and found out that the transition state has to be here between non and is peptide bond. So if it has to be here, it has to reach here, and this verifies again. So what allows this, what propels this motion and what allows it? If you remember, there were two sites. This is the whole active site, and there was a nucleotide in the middle that reached the peptide bond formation. This is nucleotide 2602, and look how many structures we have for it. In a minute I'll show you. So ribosome is a polymerase. It creates the bond and elongates it. And the question is, what is making this reaction possible? So correct positioning of AtRNA is required. An initial ptRNA has to be already flipped. How does this obtained? So if we look at the active site, in the A site there is one G here. All tRNA 3' ends are CCA amino acid, CCA, across evolution. So there is one G here to make a Watson-Crick base pair on the A site, but two on the P site. And this is also, these three of them are conserved along evolution. So this makes it much stronger, contacts that are much stronger here. So the first P site that comes in chooses, or is forced to choose this site. So now the catalytic activity, as I said earlier, is substrate positioning, nothing else, just the stereochemistry, and we think that we understood how the stereochemistry works. About the same time came a paper out of Yale University that showed, again without knowing about us, that actually the ribosome is not catalyzing peptide bond formation, but the substrate does, substrate-mediated catalysis. And they showed that when the tRNA in the P site that makes two GC base pairs is in place, and the A site, one GC in place, is in place, the OH of the C76 tRNA on the P site makes a sort of a bond or interaction with the substrate that carries the rotation, the peptide bond formation. So this is substrate mediation catalysis that comes in from very ancient times. So now the question is, where is peptide bond formed? At the A site, when the tRNA is at the A site or P site, the newly born protein is A or B or P or between. So there were lots of conversations about it and contradictions, and the ribosomologists really like that it's made in the P site and goes to the A site. And I really didn't like it. Why to do this? Why to be made in one site, go to the A site, and then back to the P site to come out through the tunnel? So we thought maybe between the two sites. And this is the work that I highlighted earlier, Massa and Carle. They looked for the transition state, and they found that with this type of rotation, it is made here. And I want to show you. So it also explains how the ribosome can be a polymerase. So here it is. If you remember, these are the two nucleotides. This is the rotation. This is the tRNA. And the one that is here in the middle can be the propeller. Here it is. This is the one. And I put here only the A site and the CCA of the P site. This is this one, this nucleotide. And as I told you earlier, I wanted to show, I couldn't, it was not in place. Here are all the conformations of it, just by having a sparsomycin, which is a very, very small molecule. It can go from here to here. And it can do much more, from here to here to here to here. Doesn't it give you a feeling of propeller? So we think that this particular nucleotide, 2602, it propels the rotatory motion. And as I said earlier, and we had the, why? Why? Why do we have to look at it this way? Oh, good. we showed it by using Sato's results, Wenke's results, and our results. So if we look now, we go into the question that I really want to ask. We look now where, in which part of the ribosome are the functions. So the decoding in the A side, in the small subunit, sorry, is here. And peptide bond formation is here. And you can see it's all gray in both places, all gray. Just everything, everywhere the ribosome functions is RNA. So it's a ribozyme, an RNA machine. And what does it mean? So when I ask kids, when I talk to kids, high school kids, even grammar school kids, and I ask them, why do you think the ribosome is an RNA machine? And they say in Hebrew, because this will prevent protectia. It means in English it will prevent bias. That there is one kingdom that makes the bonds and one that is created by the bonds. Because if it would be the same kingdom, could be that I love Emmanuel more than anybody else, I will make more of him if we are the same kingdom and so on. So this is what the kids say, and I think that they are right in a way, because this is a sophisticated regulatory machinery. It is also in good agreement with the composition of ribosomes, 2-to-1 RNA to proteins, except for mitochondria, but even in mitochondria that is 1-to-1, the active sites are RNA made and at the same place. So we think this hints that RNA existed, since RNA existed before or dominated proteins, it could be the one, but RNA is known to be a rather inefficient enzyme, and ribosome is very efficient. It became efficient by introducing proteins, and I'll come back to it. So first of all, about the symmetrical region, or the semi-symmetrical region. Its high conservation, as I showed you earlier, indicates its existence beyond environmental conditions. And we propose to call it the proto-ribosome, the before ribosome. So this means that the prebiotic bonding entity, termed by us proto-ribosome, is still functioning in the contemporary ribosomes. We still have it in us, in millions, as part of the ribosome. Or in an ancient way to say, an RNA apparatus with catalytic capabilities functions within the contemporary ribosomes. And based on the suggested existence of an RNA-dominated world, and on the finding that RNA can replicate and elongate itself and has catalytic capabilities, we propose that the proto-ribosome is not only the beginning of ribosomes, but also the entity around which life has evolved, the whole concept of life. I just want to show you the size of it in the ribosome. So what you see here rotating is the large subunit, and the piece we talk about is this little one that looks when there is RNA inside in space filling. You can see here the ribosome is in gray, and the peptide bond, as we discussed earlier, is made within it. And this machinery, the way it looks today, looks like that. Two parts, symmetrical, and in the middle of it, you can see the two substrates. You can see it now larger, two parts, like a pocket. So we suggested that it existed and functioned as an apparatus in the RNA-dominated world. Just to say that RNA can made itself under prebiotic conditions. I have here several references, neither of them written by us. So one of our hypotheses suggests that the catalytic proto-ribosome is a product of dimerization, that even then there was dimerization. And this requires the existence of self-replicating, self-folding, and self-dimerizing RNA molecules. What do I mean? Self-folding, self-dimerizing from top and from the side. And we were quite not very popular with it until several papers came out and supported it. So the pieces, these pieces can be just random pieces that should fold as stem, elbow stem, that we could synthesize as much as we want. Can be the primitive version, this will be the construct for it, or they can mimic the part of the ribosome that is within the symmetry related region, because this is completely, almost completely conserved, so we could take any one we wanted. So if we look at this, we have a P and an A possible constructs, A and P. And you see what you can already? What we can play with are these sides, so these ones. So we did many of these constructs, and we mutated them, we made many more, and many more. And what we found, and also our red, what we call red, that there is a non-uniform tendency to dimerize. Some dimerized and some did not. and we call it pre-Darwinian Darwinism, those that dimerized. And this is within the pocket, it's the P side, and within others, 50%, but we cannot say why. And I just want to show you that we see that they dimerize, that their molecular weight went on. So, as I said, only the backbone is symmetrical, no sequence symmetry. What does it mean? Maybe that in the beginning there was a homodimer and it became afterwards a heterodimer. And you can see here P on top of A, you see that they are a little bit different. So maybe it started with P region. So, and since RNA can be the template for itself, it's conceivable that the proto-ribosome was dimeric, that was made by gene duplication. Now I have a whole discussion why gene duplication is not very liked, but I will jump it. Anyway, I just want to say that we construct the proto-ribosome in many strategies, 2 segments, 2 fragments, 1 fragment, many fragments. They all bring the same thing. Some dimerize, some not. So let's jump this, and this is what I showed. Also, we don't need a compactization. It will be nice to have some type of missiles, but we don't. We don't need it. We may have. And we could show that all this, we could show in the literature, that all these structures of RNA can be made and kept in micelles. So it can be that there were some type of protocells. We are not against, but we don't really need it. Why should RNA construct a machine that polymerizes amino acid? It means create competitors better than him. So I don't think RNA sat there and decided to do it. I think that there was a pre-proto-ribosome, a machinery in the ancient times that was hijacked, that could do all types of things RNA needed and hijacked by amino acids. So to show it, ongoing binding experiments are shown, and we can bind to what we call our dimer, some type of RNA precursors. So now the question that I asked you, what was first, the genetic code or its products? Products are the proteins. So this is thought about the emergence of the genetic code. It must not be. But first of all, some other, many good RNA scientists showed that substrate could be together with activated amino acid made by RNA to RNA. Also, this See, this is the entrance to the tunnel, and this is the whole symmetrical region. Here, this part, if you can see, more shiny, is the entrance to the tunnel. So essentially, the first dipeptide could be the substrate for more and more, and oligopeptides could be made this way. And those oligopeptides that could be needed in the RNA world or helping the RNA world, they survived. They were fit and they survived. Needed mainly, let's say, oligopeptide histidine rich that can transport zinc, or those that stabilize the machine. They could be those that survive. So for stabilization, we have the same in the whole ribosome. Here is one protein that stabilizes it. Here is another one. I don't want to talk about it much, but it can be. So this suggests that the genetic code was created by or according to its product. These were found fit and useful and therefore survived, and led to the creation of a primitive original genetic code which co-evolved together with its products. So this is what I said, genetic code co-evolved with its product. Ribosome became, I'll go a bit fast, ribosome became efficient by adding the proteins. So from an inefficient RNA enzyme, it became the ribosome, and you can see here ribosomal proteins in the small subunit, the large subunit, all have very long extensions or very long internal loops, which penetrate into the structure. This is the small subunit. We talk about these 4 proteins, how they sit in here. And for instance, this one, most of the protein is here, but its extension is very long, reaches the decoding site. So any mutation here does not stop decoding, but disturb decoding, allow mistakes. And we think it does it because it changes the structure around it. So in summary, I told you, and I want to show you the structure that we talked about. Here it's in the ribosome. This is the way it looks from top. This is from the side. This is the whole machinery. And I also want to repeat that the ribosome architecture hints at its origin. This part of it, 3 to 4%, depending on the source, is probably what we think, beginning of life, before the ribosome, before life. And repeat it too, that the prebiotic chemical bonding entity is still functioning in all our contemporary ribosomes in all cells. I also can ask you, what was first, the chicken or the egg? The genetic code or the protein? So it's still difficult. Therefore, we answer this. The ribosome was first. So I want to start with credit. The first credit is to Nancy and Margaret Ginsburg that they looked at the Dead Sea. I think all of you saw the Dead Sea, but anyway, this is the way it looks from top. Dead, nothing here. But there are colonies of bacteria that were discovered, as I said, by Benz and Margaret Ginsburg, late Margaret Ginsburg, sorry to hear this. And these were the first, the ribosomes from these bacteria were the first to crystallize. And it was a breakthrough because many others did not, by many other groups too. I had 2 groups, one in Hamburg and one in Israel. Those in Israel didn't care, but those from Hamburg came all the way from Hamburg to look at the Dead Sea, to look for better bacteria beds. And here they are, having better time. So the group in Hamburg was terminated 10 years ago. Only the group in Israel is now functioning, and you can see them here. I wanted to also to thank Michael Sela and Chaim Harari and Gunter Wittmann for helping me start and keeping with me. But now I want to show you Ilana. עוד שנייה. למה צריך לראות זה ככה? That identified this. You know, it was easier to solve the structure of ribosomes than to control the light. Ilana found the pseudosymmetry. Anat is running the group, especially now when I am entertaining you. I can't talk to you about all, but I want to show you Tamar, who came to us 16 years ago for 10 weeks. She's still around. And she had a birthday the day I took the picture. Here is what she baked for her birthday. So this shows that ribosomes are considered sweet. I want to tell you that my family always supported me. My granddaughter, that is now an army girl, told everybody in the world that at the age of 5 she invited me to her kindergarten. And she gave me the best ever prize I got. This is the medal, the Nobel medal. But this is the real prize. The grandma of the year is Ada Yonath. And when I asked her which year, every prize has a year. She said, you have to reproof yourself every year. The minute you fail, I take it off the wall, so it's still on the wall. Ribosomes, you see, it's still on the wall. Ribosomes became famous and gave the inspiration for masks in Purim, this carnival. She is a ribosome, and he's me, see? And kids, even young kids. This is what happened to me. Kishka drew it without meeting me and without being a crystallographer or a scientist. And thank you. So if you let me one more minute, that I've forgotten. Kishka sensed that we will be looking for symmetry. You see, he drew it symmetrical. This and that, small subunit, large subunit, large subunit. And this is what will carry us to the next lecture about antibiotics. This is what Kishka thinks will be good. Thanks. עכשיו אפשר.
Now we can have the lives. Thank you, Ada, for taking us step by step through ribosome synthesis. Questions, please.
So, in the very beginning, you mentioned that all ribosomes are created equal.
Created?
Yeah, but sometimes ago.
I didn't say that.
No?
They are working equal.
Okay, but there was a paper that shows differential stoichiometry.
Okay, you're right. I didn't say they were created.
Okay,
I even didn't say that
misunderstanding.
they keep the same stoichiometry.
Sorry.
I would have said it 10 years ago, but now we know much more.
Okay, sorry, misunderstood.
All of them doesn't matter how they created, and the stoichiometry and all this doesn't matter. It does not change the pseudo-symmetrical region inside this.
Question I want to ask is, how do you comment that this differential stoichiometry can have regulatory role?
Next time, next full hour lecture. It's not just a comment. It's to show one by one what one component is doing and can be done otherwise or not needed.
So just a question. I know that you probably don't have a real mechanism to give, but how would you imagine would be the evolution from the ribosome, from the RNA world, and the proto-ribosome to what we have today?
Well, this I can answer you partially. First of all, there were these little occasional oligopeptides that were loved and stuck. And then the machine became better and better functioning, and life before life became more complex, so more proteins were needed, and so on.
And only at that point did the adapter come, after there was already the existence of it.
After the small ones, yes.
Mm-hmm.
But at which point you're asking too much?
Thank you.
Excuse me? Could tRNA bind mRNA before entering the ribosome? Could tRNA bind mRNA before entering
Binds
the
the
ribosome?
amino acid. The amino acid it binds before entering. If it binds to the messenger before it goes into the ribosome, that's a fantastic project, which we tried for 2.5, 3 years, and we couldn't show here or there. But you are young. It's fantastic, because even in the little movie, all the ribosomes are standing nicely in line and each goes in. There are 20 types of, all the amino acids, 20 types of amino acids. And maybe some of them are with some copies, or hundreds of them, all standing in this little entrance, and there is no way to go out from structural point of view. So what happens? How do they know to stand there in place and in time? This is what we wanted to answer. And the hypothesis was that they bind earlier, or at least they recognize earlier the messenger. We couldn't show it, and unfortunately not yet. But it's very intriguing. And I think all of you heard about Aaron Klug from MRC, Professor Dr. Sir Aaron Klug. He looked at this movie and he said, Ada. You know, he was very attached to Beersheba University, to Ben Gurion. Ada, you are showing people standing in line like in Cambridge, not like in Beersheba. In Beersheba, they would be hitting and banging. So that's the way he thinks about it, and he's right. And we try to show that they're standing in line, because otherwise it couldn't be. If the ribosome stopped every time he found a mistake, it won't be able to make 40 peptide bonds a second, not even 5, not even 1. And there is no way for going back, just to get rid of everything, start from scratch. This happens. We know that it happens. We know. I showed the level of mistake, and I didn't comment, but I showed it. It makes many more mistakes in the decoding. We could show that if, not us, but scientists show, if they take only the small subunit, it makes mistakes, maybe 1 to 100. But it has such a good proofreading machinery that it gets rid of the mistakes before we even can identify them. So we see 1 to 1,000,000, but it makes more. But this is before it is a ribosome. Once it is a ribosome, it cannot play this game. There is no way the tRNA can go backwards from structure point of view.
The tunnel through which the polypeptide comes out, whether it has the same conformation or it changes its conformation depending on the amino acids?
I really didn't understand the question.
So that there is a tunnel through which the...
There is a tunnel.
Yeah. So depending on the different amino acids, does the tunnel size change or it just keeps the same?
It's fantastic. You have a very great group of people. This is a question that many scientists are trying to answer for a long time. Once they agree that there is a tunnel, which took about 10 years, but once they try to show it and to try to follow different sequences, biochemically, by electron microscope, by FRET, and there are several ideas, but neither of them was shown fully. You're also young. That's a very intriguing question.